Get ready to blast off into the incredible world of brain-computer interfaces. Neurocareers doing the impossible is taking you on a journey to meet the fearless pioneers pushing the boundaries of what's possible. In this special series, we'll be shining a spotlight on the nominees and winners of the International BCI Award, one of the biggest and most prestigious awards in the BCI world. You'll hear from BCI professionals as they share their revolutionary work and get a behind-the-scenes sneak peek at what it takes to be a winner. So, buckle up, grab a snack, and get ready to be amazed as we explore the impossible becoming a reality in the world of BCIs. Before we blast off, I want to give a big thanks to the co-host of this BCI Award edition on our podcast, Dr. Christoph Guger and GTech Medical Engineering. When I was a faculty member at the University of Cincinnati and Cincinnati Children's Hospital Medical Center, my team and I did some pretty amazing stuff for patients needing epilepsy surgery. We would put special sensors, also called grids, directly on their brain to prepare for surgery and create maps of their brain in a much faster and safer way than is usually done. It's called high gamma mapping, and it lets us figure out in just a few minutes the essential parts of the brain that need to be spared during surgery. Can you imagine? You can create a map of language, motor, and even math processing in no time. It was the first time this innovative technology was used at Cincinnati Children's Hospital Medical Center. When I moved to Florida, I established the first functional brain mapping and BCI program at Florida Hospital. I continue to use high gamma mapping to help epilepsy patients avoid losing their ability to speak or move after surgery. But even cooler than that, I created brain-computer interface studies that let patients control things with their brain in real time. Our patients could even spell words with incredible speed by just using their brains. No hands involved. I also discovered that brain-computer interfaces could help patients move their chronically impaired hands or legs years after a stroke when not much else could help them. So, I started working with the faculty from Advent Health University to help these patients restore their ability to use their hands. I received special training for it, and it's still mind-blowing how cool it is. My favorite part of what I do is teaching. At my established Institute of Neuro Approaches, I've integrated all my experience and knowledge into a unique course on brain-computer interfaces. With graduate neurobiology students, we started with theory, and then I gave them the BCI equipment so they could have hands-on experience working with BCIs. It significantly improved the way students learn, and they absolutely love this practical part of learning how to use neurotechnology. And all of this was made possible thanks to GTech's awesome brain-computer interface technology. They have everything from high-tech up to 1024-channel EEG systems to wearables, tools for neurorehabilitation, such as the medical grade system recoveries and educational kits with unicorn hybrid block for learning how to work with BCI technology. But the most important part is their support and care. I have enjoyed working and collaborating with Dr. Christoph Guger and GTEC employees for over 15 years, and I hope for many more successful years ahead. So, if you are interested in GTEC's brain computer interfaces and neurotechnologies, check out their website at gtech.at. It's worth it. Ever wondered how we can decode fast limb movement using brain-computer interfaces with slow data acquisition rates, like, for example, in optical imaging? This challenge has long puzzled researchers, but Seungbin Park and her team, led by Dr. Maria Dadarlat at Purdue University, have come up with a groundbreaking solution. In this episode of the BCI Award Neurocareers podcast series, we tackle the problem of low sampling rates in decoding limb movements. While traditional methods struggle with capturing fast limb trajectories, Seungbin Park and her colleagues have used a deep learning approach that overcomes this barrier. By employing a recurrent encoder-decoder network, they were able to decode rapid limb movements of a running mouse using slow two-photon calcium imaging, a major breakthrough in minimally invasive, optically based BCI for multi-limb control. Join us as Seungbin shares how their innovative approach earned them a spot as one of the 12 finalists for the BCI Award 2023. She'll also offer insights and advice on how to make a successful BCI Award submission. Tune in to hear Seungbin Park's incredible story of pushing the boundaries in neurotech. Hello, Seungbin. It's a great pleasure to have you today on our podcast. Can you please introduce yourself to our listeners and tell them where you are joining us from what part of the world?
Hi, nice to meet you. I'm Seungbin Park, and I'm a third-year PhD student now, about to start the fourth year, majoring in biomedical engineering at Purdue University with Dr. Maria Dadarlat. Here I'm working on applying deep learning to decode neural signals recorded by two-photon calcium imaging. Also, I started a project to analyze the dimension of somatosensory information encoded in the somatosensory cortex areas using dimensionality reduction methods. I was also trained to perform neurosurgeries on mice and use two-photon microscope for imaging, but recently I'm more focused on computational work.
Yes, very interesting and very interesting career path. So you trained yourself to do neurosurgery, and now you are switching more towards computational approaches. So I am very interested to explore all of that. I want to ask you to briefly summarize your work that was nominated for the BCI Award in 2023, which is called Towards an Optimal BCI, Overcoming the Limitation of Low Sampling Rate for Decoding Limb Movement. If you can just give a brief summary, because later we will go into more detail, but to start with.
Okay, thank you for giving us opportunity to introduce our work. Like you said, the title of the project is Overcoming the Limitation of Low Sampling Rate for Decoding Limb Movements towards an Optical BCI. The main aim of this study is to decode these neural signals recorded by two-photon calcium imaging to predict the limb movement of a freely running mouse. We believe that this study can contribute to the development of the BCI, especially the next generation of optical BCI.
Thank you so much. And now I would like to take a step back. a little bit on your career development. How did you get to this point when you are doing this amazing study, trying to decode and predict the movement of really a moving rat, and you are working with this amazing technique? Did you always know what you want to do when you were growing up? How did you come to this place?
I feel like I would say I didn't know. I don't know. I don't know what to do or what I wanna do. If I briefly introduce my path, first I started research, majoring in computer science for my master's degree before my PhD. And there I worked on applying deep learning to various types of biomedical data. For example, I applied various machine learning algorithms to diagnose some surgical parameters for vision correction from patient clinical data. Or even I also built a neural network for the segmentation of bones from head CT scans. So it was not technically neuroscience field, but after I finished my master's degree, before I decided what to do after my PhD, I thought I want to work in BCI field because it was my dream since I was young to develop technology that can help paralyzed people to move or communicate with the world. So that's why I guess I changed the field slightly and I joined Dr. Maria Dadarlat's lab and started my PhD program. Based on these experiences during my master's degree in deep learning and coding, so that's why I'm working on applying deep learning to the data collected in our lab or any other computational works.
Thank you so much. And you mentioned that actually you had a dream to work with BCIs, and that's why you got into this field. Do you remember the time when you first time learned about BCIs? What was it?
The first moment I started to have interest in neuroscience is that, well, first I remember the first time I learned about neuron and I thought, wow, it's really fascinating. I think it was when I was like 12 years old, 11 years old. I think I started to think, oh, I want to be a scientist in the future. So yeah, I thought that how neurons work or how brain works is really cool. So there was a movie or Japanese anime I really like. It's called Full Metal Alchemist. And the main character there have a robot arm and robot leg. And after he lose his leg and arm, of course he would have felt devastated. And there he gets that robot arm and leg and he starts his adventure. And I don't know why I was so into it, but I thought, wow, I want to be part of that technology, to help people who has lost their part of body or people who get paralyzed and they cannot move. I thought I want to in the part where I can help them to enjoy their full life.
Yeah. And have to help them to have this high quality of life.
Yeah.
It's so interesting to see what can inspire us. Yes, an animation film. You never know what in the world, in the surroundings, can really inspire us for all our future work. Yeah, that's a beautiful story. Thank you. My next question is now your interest in computer science specifically, and you were talking about working with neural networks, yes, with the machine learning. Why did you choose this particular field? You didn't go into engineering, for example, but you decided to approach everything from this computational perspective. So, can you maybe talk a little bit about that?
The reason why we started applying this deep learning to your data is because of the challenges related to the task. And I guess the biggest problem in the task is that the relation between this neural activity and behavior, like for example, movement, is really complicated. And even though conventionally linear method like linear regression has been used successfully, but we can expect that the relation between neural activity and the behavior is highly complicated and non-linear. So that's why people are more focusing on this non-linear method like machine learning or deep learning, considering the complex relation between the signal and behavior. And also, there are challenges related to this optical imaging. The optical imaging method we are using is calcium imaging. And the calcium imaging measures neural activity indirectly by measuring the calcium fluorescence signals in the brain. And because there is a decay time of that calcium fluorescence, there is a difference between real spikes and the calcium signal. Of course, that relation is non-linear, and it's also a big research problem to get the real neural activity or real spikes information from calcium signals. And because their relationship is complicated and non-linear, that's why we thought maybe deep learning might be a good approach.
Absolutely. Thank you. And very good that you started talking about calcium imaging. I'm sure that there are people who are not aware of this type of imaging. I'm also personally not very well familiar with it. And maybe for those of us who are not familiar, you can talk a little bit more about this type of imaging and even how it is performed from the technological point of view. Yes, how do you perform it in animals? And of course, the main principle behind it.
I will start from the method that you or other BCI people know well. Like the conventional method for BCI is, recording neural signals by using microelectrode inserted in the brain. That's one of the conventional approaches among other various methods to record neural activity. However, this approach might have some limitations because it's highly invasive because you have to insert electrode into your brain. Also, it might have a short lifetime because the electrode can be physically damaged or there might be some immune responses in the brain to the implant. Also, it can only record the activity of neurons that are especially close to the electrode. So even though there have been advancement in the technology of recording, they can record like hundreds neurons at the same time. But because behavior is related to activity of large population of neurons, like in neural circuits, it's important to observe or record large population of neurons at the same time. And compared to the approach using electrodes, optical imaging enables recording larger areas in a single cell resolution. And this is possible because this is available by introducing indicators to the brain. For example, calcium imaging records neural activity indirectly by measuring fluorescence signals emitted by calcium indicators, which can be genetically encoded or injected. So, you insert some types of proteins that can emit fluorescence when it binds to calcium. And so that's how we can measure the calcium concentration changes in the brain using a microscope. That's how optical imaging works.
Thank you. You already mentioned that a part of what you do is neurosurgery. And you particularly trained with the neurosurgery, and then you moved to more computational approaches. So, can you tell a little bit about your experience learning? How easy or how difficult was it for you to learn? How much time did it take? And what was the most challenging part for you to learn to do the neurosurgery on mice? Especially mice are very small animal. Yes, so you really need to be, I think, very careful with how you perform everything.
Yes, to be honest, I had a difficult time with animal experiment. Even though I was trained in our lab to perform those surgeries and experiments, I think I wasn't that good. I think you have to be really skillful because the quality of the imaging is highly dependent on the status of the window. You have to be really accurate and precise, and the window has to be clear for imaging. And of course, there might be some infection or problem in the mouse after surgeries. I think I wasn't that good with it. And of course, there are many people who are really good at animal experiment and who are highly interested in it, because even though there might be some ethical problems with animal experiment, but I think it's also true that animal experiment is necessary for the development of the technology and research and science. But I realized I think that's the animal experiment is not for me. So that's why I gradually changed from animal experiment to computational work in our lab based on my previous experience before I started the PhD. It's also related to the advice that maybe I can give to other students who are looking for advice. But when there are some undergraduates who ask for advice on their career, I say I highly recommend to try internships or any chances that you can experience the field, because you don't know if you're going to like it or not before you really try it. So I think it's important to take internships or any other chances that you can experience the work and see if you're good at it or if you're gonna like it, because especially animal experiment, it's really really difficult. So I think it's better to try it before you really start your job or PhD. And also, I think being patient is important with the animal experiment, because many things might not go as well as you expect, and it might take some time to see results. So I think being patient and the attitude of enjoying the process and enjoying learning would be necessary for the work or any respect.
That's a great advice. That's very important advice. And indeed, I witnessed so many cases when people wanted to go to certain fields, even speaking broadly to medicine or to nursing, and then just getting some exposure to the field, they understood that actually it's not what they want to do, and they switched to completely different areas. Yes, so thank you very much for mentioning that. And of course, people also have questions: how do I find those internships? What would be your advice? How would you advise to approach people, where to search for those internships?
First, maybe looking around where you are would be nice, like looking for chances around you or in front of you. Like, for example, if you are in a university, schools already have a lot of sources like job fairs, or there must be some websites that shows internships. So I would recommend to look around what you have for now. You can even just contact to the lab you are interested in your university, or talk with professors or people in the lab to see those opportunities. And I guess outside of where you are, I think LinkedIn might be a good start, because a lot of these job or internship opportunities. And even if you are interested in certain fields, and if you are interested in someone's work, you can just contact to them. And it's highly possible that you don't get any responses too, because people are so busy. But I think in research area, I think it's quite general approach, and I think many people might reply you back like kindly. So yeah, I think those are the approaches you can take to find internship opportunities.
Yes, I agree with your approach. Thank you. Great advice. Now, let's get back to our topic towards the optical BCI. So, you already explained to our listeners what is this calcium imaging, and now uh, how do you want to use it for brain computer interface application? What is your vision of how this can be done?
Yeah, that's a good question. And considering this optical imaging requires introducing indicators to the brain, or even though it's less invasive than electrode, it requires partial invasiveness for optical access to the brain. Also, it would need imaging system such as microscope. So it sounds like, considering all these facts, it sounds like a clinical application might not be available in the near future. And I think there are some people in BCI field who thinks that maybe optical BCI cannot be applied in real life. However, they are having numerous studies to improve related technology, such as developing small imaging systems that can be mounted in the head of freely moving animals. And furthermore, this optical imaging can also be valuable in basic and preclinical studies to understand how neurons work at the populational level and deal with challenges related to real-time signal processing, especially when the dimension of the data rises with the increased number of electro channels to record a large number of neurons. So even though application in clinic is difficult right now, but I think there is still some potentials with a lot of advantages, and I think it's also valuable in basic studies too.
Oh, yes. And it's very good that you are developing this technology. You are obviously addressing certain limitations of the technology. And this is what your project is about. First of all, about the application, you are trying to apply this technology to decode limb movement. So, can you tell a little bit more about that? What type of movements do you want to decode? Why is this important? Yes, and how it can be applicable to brain computer interfaces maybe in the future.
When we record neural activity of a mouse, first, I guess I need to explain how this animal experiment works. So after we do all those surgeries, then we can put the mouse on an airlifted ball and fix its head so it can run freely on the ball while we image its neural activity on the ball. And at the same time, we record its running using cameras. Like currently, we are using two cameras on the right side and left side. So now we have that videos of mouse running, and we can extract the coordinates of limbs in those video frames. We use a library called Dadarlat. It's also a deep learning based pose estimation library for animals in the lab. Now we have these coordinates of each limb, all four limbs in those video frames. And the aim of the study is to decode those limb coordinates or trajectories of the limbs from two-photon calcium imaging. Decoding this neural activity to predict this movement information, I think it's essential, or it's necessary to BCI application, because BCI is a system where you can control external device using your neural activity. So for that aim, you need to decode neural activity like related to movement. So I guess that can be the first aim of this application. And also, these limb coordinates can be interpreted as the proprioception, which is the sensation of our like position of our body. And if you lose that sensation, you lose a large portion of movement control. And so this sensation is also important for movement. So I think this decoding limb trajectories can be helpful to understand how this proprioceptive information is encoded in the brain, or it can be also used to provide this sensation information to BCI, because besides proprioception, for example, you also need touch to control your body. So there are people are developing a closed loop BCI where you can provide sensation information to the BCI to advance the quality of the movement of the external device. So yeah, by decoding this sensation information, you can provide it to the BCI system to help the quality of the movement.
Very good. And now let's talk about what works well in your experiments, because then we will go into challenges and specifically already towards your study that addresses these challenges. But first, let's talk about what works well in your experiments, what you can decode and fairly successfully.
The aim of this research is to decode these limb coordinates in video frames or from those recorded two-photon calcium imaging. But first, maybe I want to introduce challenges related to decoding this calcium imaging. Like I told you before, first, the calcium signal decay is relatively slower than real spikes. So the relation between underlying neural activity and the fluorescence signal is indirect and nonlinear. Furthermore, there is a trade-off between the size of the field of view and temporal resolution. I told you that you can record larger number of neurons with this optical imaging, but it might result in lower sampling rates in imaging compared to the behavior. For example, in our case, the sampling rate of this two-photon imaging is about 8 hertz, while we record the behavior of running mouse at 30 hertz to capture the accurate trajectories of limb movements. So, considering these challenges, we tried this deep learning based approach to decode limb movements from calcium imaging data, which has a lower sampling rate than limb movement signals. And by using an encoder-decoder with LSTM, we showed that the movement of all 4 limbs could be decoded from calcium imaging records from a single hemisphere.
I'm just very curious because I've never seen the actual data that you're analyzing. How does it look like? What are you analyzing?
There is neural signal and movement data. So calcium signals recorded by two-photon calcium imaging. But when you record the brain with using this two-photon microscope, you'd get these 2D fluorescence images in time series. But we run some processing on it, and then you can extract these fluorescence traces of each neuron. So as a result, you will get this 2D matrix, which has the size of the number of neurons and the length of the record. It's like the fluorescence traces of each neuron. So that's our neural signal. And the behavior data, movement information is this limb coordinates of a mouse in a video frame. Does it make sense?
Yes, yes, it does. Yeah. Now I have a good picture of that. Very interesting. How many neurons are you recording?
We can get like thousands. And for example, in our one subject, we could get like 3,000 of neurons.
And you mentioned the challenges, and obviously you are addressing them. And I think that's one of the things that you did in your BCI Award nominated study. Can you talk a little bit about that? How are you overcoming the limitations, especially of this low sampling rate?
Because two-photon microscope has low sampling rate, that neural signal, which would be input to the decoding model, is shorter than the output, which is the limb trajectory. For example, 5 neural frames will correspond to like 20 behavior frames. So, it's like you want to get more information from a small amount of data. So it's like expanding the sequence, like from small sequence to longer sequence. But we used this encoder-decoder architect. So LSTM is a well-known type of neural network. It's a type of recurrent neural network. And this LSTM is widely used for time sequence data. And we have this separate encoder LSTM and decoder LSTM. And because they are separate, we can set the length of them differently. So that's how we can set the input and output length differently. So that's how we approach to this problem of different lengths of sequence of input and output.
Yes. So, what are your results? What do you find when you're applying this approach, this technique to your data?
First, we showed that the model can decode at a certain level. So it means it could predict that limb trajectories close to these true limb trajectories from the recorded neural activity. And because it is generally known that like one side of the brain controls the different side of the body, like it is called contralateral side. But we also confirmed that even though there's a main limb among four limbs, so there's one limb that model can decode very well. But even though the decoding performance is lower, we showed that all four limbs could be decoded from one single hemisphere record.
Very interesting. And you mentioned that there is one limb, which movements are being decoded best. I'm just curious, what is an explanation for that?
Because it's well known that there's somatoping map that specific areas of brain controls some part of the body. So I think in the imaging area there's one like main limb or controlling limb to that imaging area, but I think the result shows that besides those main limb, other limbs, like information for other limbs or movement of other limbs are also encoded in those imaging area. Even though it's generally known that one part of brain controls the contralateral body, but there have been other studies showing that one side of the brain also has information for ipsilateral body. So I think it aligns well with other study results.
Mhm. So, if you would summarize the main results of your study, what would those be?
I would say our approach showed that the movement of all four limbs could be decoded from calcium imaging records from a single hemisphere.
And what does this mean for the future development of the technology?
I guess the fact that you can decode all limbs from a single hemisphere is beneficial because then it's easier to record part of the brain than all of the brain to control all part of the body. Also, I didn't mention it so far, but the neural network could pick the important neurons to decode this movement. But I think it's also well known in neuroscience field that neurons coding in the brain is quite sparse. So there are just some really important neurons among a lot of neurons that is highly correlated to movement or important to movement. So I guess if you consider that, then you only need to focus on some part of neurons or a few neurons to decode movement. So I think that can decrease the cost or computational work to decode movement for BCI.
What was the most challenging part for you in all this project?
Most challenging part was that when it didn't work. It didn't work when I first tried. Like when I tried the simplest model I can think about and the simplest processing steps that I can apply to data, and it didn't work. I felt really frustrated, but I think my PI, my PhD advisor, Dr. Maria Dadarlat, helped me a lot. And by the way, I want to take this as an opportunity to express my gratitude to her because she's always inspiring and encouraging. Like every time I or students in our lab feels frustrated because results are bad, she give us insight to analyze the result and make that as an opportunity to go to next step. So for example, in my case, when it didn't work, Dr. Dadarlat helped me analyze why it didn't work. And I found out that I had to do more processing on that fluorescence traces because like I told you, it's a noisier version of spikes. So for the decoding, less noisier data was needed. I still remember the moment when I tried like the process data and it worked. So I think it's important not to feel too frustrated and not to give up and just try to think about why it didn't work and change it and try it again and see it works.
That's also a great advice. Thank you. Thank you for that. The next question is, we always hope, yes, to find something. We have some hypothesis, we have some preliminary ideas of how things might work. But sometimes we might find something that was unexpected. Yes, something that was surprising while we are doing our study and analyzing the data. Was there anything that surprised you that was unexpected in the study?
Would say I think it's most of the time because I think it's a characteristic of neuroscience research field too. Because in neuroscience field, people are trying to understand how brain works and try to reveal what's unknown for now. So when you run some experiment and run some analysis and see results, it might be unexpected or it might be opposite from what you expect. But again, my advisor, Dr. Maria Dadarlat, always say we have hypothesis and we expect this result A, but she always say it doesn't have to be A. It can be B or C or something completely different. And I think that's the nature of this field of neuroscience.
Mhm. Maybe there was something that you learned during this project that not only related to what you were working on, but something about the brain, something about the neuroscience that you learned along the way.
Well, I guess that would be analyzing this importance of neurons from neural network. Well, besides this decoding performance, other aspect of our research was analyzing how this neural network worked. It's also called explainable AI. So we analyze this importance of feature, which is neurons here, and we quantified like which neurons were used importantly for this decoding in this neural network. I already briefly told you the result that they showed that like highly important neurons were just sparsely distributed over the space. But first, we expected that those highly important neurons would be like clusters in the specific areas because we can record large area of the brain. The imaging area included like some of the somatosensory cortex and some of the motor cortex. And, for example, if motor cortex is a main controlling neurons in this movement, then those um, neurons in specific motor cortex area would be important for the decoding. But the results show that no, they were just dispersed in the space. But we also thought that makes sense because sparse coding in the brain is also well known. So I think that was the interesting part I experienced.
Yes, yes, it absolutely is. So, what are your next steps? Where are you going from here?
We thought our next project would be decoding these raw calcium images, not the processed extracted fluorescence traces, because I told you earlier that we use these processed extracted fluorescence traces for of each neuron from 2D image. But of course, for real time application, you cannot have time for processing the data. It's better to use raw data right after you record it, right? So our goal would be try this decoding to raw 2D images from two-photon calcium imaging.
Yes, very nice. Thank you. And now I would like to talk a little bit about your BCI Award submission. Why did you decide to submit your work for BCI Award?
I want to take this opportunity to say thank you to my advisor, Dr. Maria Dadarlat because she highly encouraged me to apply for this. But I want to say, to be honest, at the beginning, I hesitated to apply for it because I felt like there are so many good BCI works out there and no one would care about what I'm doing. So also I had no idea that our work would be nominated. I had zero expectations. And especially since this concept of optical BCI is relatively new. So, compared to conventional approach using electrodes to record neural signals, and especially there are many studies for human subjects. But I think the BCI Award Nomination Committee highly valued the aspect of innovation because which is essential to change any field of research. So yeah, I hesitated, but I did. So if I can give an advice to people who are interested in BCI award, I want to say just be confident and just do it, try it. Because even though you feel like your work is trivial, these self-doubts are not true. So I just want to say, try it.
Very good, very good. Thank you for mentioning the innovation as one of the main criteria for the award. If you would summarize very briefly, what was the main innovation of your specific study?
Using optical imaging for BCI, I think that's new to BCI field.
Mhm, mhm. How do you see the development of this technology in the ideal world? Yes, if everything goes by the plan and we have all the tools, everything we need to develop it, how would this technology look like in the future?
More work is needed to get to that point, but I guess if like all the technologies are good enough, I guess instead of inserting electrode into your brain, which might physically hurt your brain, you can just have a small window on your brain, and you can just put really small imaging system on your head, and you can use that optical system for recording neural activity to control the computer or device, or even you can stimulate neurons with this optical system. It's one of the advantages of optical imaging that you can record and stimulate neurons at the same time. So yeah, I guess in the future, you would have small window on your brain, and you can just put the imaging device, or you can just remove it easily, and you can get neural signals from your brain. And at the same time, if you need any therapeutic stimulation, then yeah, you can easily stimulate those neurons too.
Mhm. And how long this system can function? For example, right now in mice, for how long this system stays in place and still provides good data?
It depends. Usually, if you've performed the surgery really well, it can last like months, and it can just last until the mouse lifespan is done. And it's also like another big research goal to make it more stable.
Mhm. Maybe you can just provide a brief perspective in terms of what is going on in the world with this particular technology that you describe, yes, with calcium imaging. What are other approaches, what are other labs are trying to do?
Like I said, maybe it might be a little bit far away from real application for now, or it might be difficult to think that it can be used in real life application, and especially for human. But it has so many advantages that you can record so many neurons at the same time, and also you can use it for stimulation, and it's even more free from that electrical stimulation artifact. So with all these advantages, I know there are many labs who are using two-photon calcium imaging for this research purpose, like basic and preclinical studies to understand how brain works and to develop the better BCI technique using that experimental data.
Thank you. And a little bit about your career. My question is, what was the most challenging part of your career development up to this point, and how did you solve that challenge?
I think most challenging moments for a PhD student would be like when you get bad results and you feel like you cannot make it better, or you feel like you're doing something wrong. That's why I also said I think patience is really important, because it's a long journey. If you start PhD, it can be really long, and it might take some time to get results. And there must be some moments you feel you want to quit, or you feel it's tough. But the important thing is to just go through that moment and just do it and keep pursuing. Yeah. So I think patience is important.
Yeah, thank you so much. That's very good advice. And what are you planning on in the future to do when you finish your PhD work? Do you see yourself more in academic settings? Do you see yourself more in industry? What interests you and what are you planning? At least at this point, it might change, of course. Yes, but at least what do you think now?
Yeah, that's so true. I keep changing my mind so many times. Like you said, I'm hoping I can graduate next May in 2025, next year. And I've been actively looking for a job after my PhD. I think I'm more interested in industry for now, but I am open to a wide range of future opportunities. I think I like research, so I'm interested in postdocs too. And even my interest in the research field is quite wide too. And of course, I'm interested in BCI field, but recently I'm also interested in drug development using this deep learning. Also, I used to work on this analyzing medical images using deep learning. So those are fields I'm interested in. And I think my main interest is applying this deep learning, artificial intelligence, machine learning to in the biomedical field, to develop the technology that can help people and improve people health.
And what do you think are the main skills that helped you to work on all these projects and get the results?
I would say my research field has been really various and wide. I feel like I did everything a little bit. But I think the main thing you learn from graduate school or your PhD is how you process the problem you face in front of you. The process of how you solve a problem is the important aspect you learn from graduate school or PhD, no matter which field you are in.
Mhm. And you also mentioned machine learning, and this is your interest, you want to apply it. How did you learn about machine learning and machine learning techniques? Were these the courses at your university? Did you also try to study additionally, took some courses? So maybe you can share experience with those who actually want to learn more about machine learning and get into the field.
Yeah, I think lectures can be a good start because, especially these days, artificial intelligence is really hot topic. So I guess most of the university have the lectures. But for me, I think the best way to learn the technology is you try it for yourself. Like you try a project on your own. And there are so many good sources out there in the internet. But I guess to begin, I usually highly recommend the course on Coursera. There is a really popular, famous deep learning course by Andrew Ng in Coursera. I really like that lecture. Also, there's a famous Stanford lecture for deep learning and computer vision. But besides this, I bet there are so many good resources, lectures on the internet and in your university. Oh, and after you learn the concept and understand the concept, how this deep learning or machine learning neural network works. Like I told you, it's important to try a project on your own. And to begin, I highly recommend the PyTorch website because they have a really good documentation and they have really good examples with all the source code and example of data. So I think it's a good way to start to follow those example codes.
Great advice. And maybe we can add this information later into our podcast notes so that our listeners can find and PyTorch and also those courses on Coursera and at least to get a start in this area. Excellent. Thank you so much. And a question that I started asking our guests recently, maybe you are aware of some misconception in neuroscience and neurotechnologies that you witnessed. Yes, so we have some of the misconceptions, let's say, that people already believe that we can read human mind, yes, or we can influence people by stimulating their brain. Yes. So there are various misconceptions, and I'm wondering maybe you witnessed some of those, and you can share this misconception with our listeners and explain why it is true or not true.
Yeah, I think I've been reading, some comments online saying like as a joke or as being serious, like, people can control other people's mind by like stimulation. First, I want to say I'm not like super expert, but I want to say technology is not that level. Like, you know, technology cannot just easily manipulate someone's mind. Technology is not there yet. But I think I want to say to people that more focus on the good result, good effect, because there are so many good therapeutic technologies using this brain stimulation. Like, for example, deep brain stimulation, as I know, they are already well used in clinic. They can be used for therapy for depression or autism or to control like any other pathology. And yeah, when I see those therapeutic technologies, I feel really admired because these technology is being used in clinic, and this technology can have like less side effect and can have more effect. So I want to say there are good therapeutic technology in clinic too. So I want to say to people to focus on those good sides.
Thank you so much. And as we're nearing the end of our podcast, you can say something to our listeners, provide some recommendation, advice, anything you want to say to them.
Like I told you in my BCI Award application, I just want to say try it. And, as many as experience you have, you know what you like and what you dislike. And, it helps you decide on big steps on your career. And yeah, so I just want to say try it and with the positive attitude, like my advisor, Dr. Maria Dadarlat.
Yes, thank you. Great advice. And even in spite of the doubts, yes, like you did, you were doubtful, but in spite of that, you did, and your study was nominated for BCI Award, and now you have a chapter that will be coming out in a book, and now you have a podcast, and I'm sure it will help in your future career development because it is an acknowledgement from established people in BCI field. So I really admire you for doing it in spite of all the doubts you had.
First, again, I want to say thank you to giving us this opportunity to introduce our work. And I want to say thank you to BCI Award to give us opportunity and nominate us. And also, I want to say especially thank you to Megan Lipton, uh, PhD student in our lab. And she was the one who mainly worked on animal experiment. And, she was responsible for the animal experiment, and the project would not have been possible without her work. So I want to make sure that her work can be acknowledged here.
Yes, of course, of course. Yes, so we're acknowledging Megan Lipton and your lab and your advisor. So thank you very much for doing this beautiful work, very promising work, and I would love to see how it continues and follow up on this. And of course, I wish you all possible success with your studies, and maybe in several years we can resume and see where you are at and what other amazing things you are doing. So thank you very much, Seungbin. It was a great pleasure.
Thank you so much. It was a pleasure to talk with you too.
Are you a fearless pioneer in the world of brain-computer interfaces? Do you have an amazing project that's pushing the boundaries of what's possible? Then we want to hear from you. The International BCI Award is one of the biggest and most prestigious awards in the BCI world, and the deadline for submission is September 1st. But don't worry, you don't need a top-notch budget or equipment to be a nominee or a winner. All it takes is a great idea and the determination to make the impossible possible. So, submit your project now at bci-award.com or find the link in our podcast notes and join us on this incredible journey to explore the world of BCIs. And make sure to listen to our upcoming BCI Award Neurocareers podcast series for tips and tricks on creating a successful submission. Who knows? You might be the next nominee or winner of the International BCI Award and proudly showcase your nomination on your resume to impress potential employers and establish a successful career in neurotechnologies. But don't stop there. If you are looking for more guidance on succeeding in neurocareers, book a free consultation with me, your podcast host, Dr. K, at the Institute of Neuro Approaches. So, what are you waiting for? Let's make the impossible possible together.